Hello friends and viewers. In this video, I'm going to talk about the immunological MHC1 and MHC2 structures and some other features. As we know that in our body, there are two types of cell mainly. One is the non-immune cells and another one is the immune cells. So in the non-immune cell, there are different types of cells like lung, pancreas, heart, and so on. For the immune cells, there's cells called APC or antigen presenting cell. These are mainly the macrophages, dendritic cell, and B cell. Those are called professional APCs, and these APCs express the MHC2. But all cell, like non-immune cell and immune cell, all express the MHC1. So let's go to the structure of this MHC1 and MHC2 molecule. Those are very important for the antigen presentation. So in the left side is the MHC1, in the right side is the MHC2. So for the MHC1, this is the cell surface of the cell, and MHC1 has one big chain. This is the C terminal of the chain and this is the N terminal of the chain. And in this chain, there are three domains. The domain 1 is the alpha 1, alpha 2, alpha 3. Within these three domains, there are alpha 1 and alpha 2. That part make the cleft where the peptide are bound here to present to the T cell. And if you look from the ribbon structure of the MHC1, this looks like that. Alpha 1 is here, alpha 2 is here, and alpha 3 is here. And thus another part of the MHC1 is the beta 2 microglobulin is attached here with the alpha chain non-covalently. And these are the disulfide bond. Compared to the MHC1, if you go to the MHC2, there are two chains here, in the right one and the left one. Each of the chain is divided into two domains, 1, 2, 1, 2. The right side of the chain called the beta chain and the left one is the alpha chain. And for the MHC2, the peptide binds with both the alpha and the beta chain, where they make the cleft here. If you look at the ribbon structure of the MHC2, this is the alpha 1, beta 1, alpha 2, beta 2. In addition, the MHC2, both chains are inside of the cell membrane. There are some specific features of the MHC1 and MHC2. First of all, MHC1 found in all nucleated cells, but MHC2 are only found in the antigen presenting cell. In terms of the peptide presentation, MHC1 present the endogenous or inside of the cell peptide, and MHC2 present the exogenous or outside of the cell peptides. Who recognize the MHC? The CD8 cytotoxic T cell recognize the MHC1 in addition to the peptide. On the other hand, CD4 T cell can recognize the MHC2. in addition to the peptide. In terms of function, MHC Class I help to kill the infected cell through the CD8 cytotoxicity. On the other hand, the MHC2 can coordinate different types of function like cytokine production, B cell health through the CD4 T cell. Since these are the protein MHC1 and MHC2, they have the genes. So these are the HLA A, B, C are the genes for the MHC1, and the HLA DP, DQ, DR are the genes for the MHC2. The HLA meaning the human leukocyte antigen. So there are some interesting facts about the MHC1 and MHC2 beyond the immunological features. The MHC is like a fingerprint. As you see, the fingerprint is different from person to person. MHC are different from person to person. That makes very difficult for the organ transplantation. Even the sibling may not match for the organ donation. Interestingly, MHC can affect who you are attracted by. Studies in animal found that different MHC gene containing animal are attracted by each other rather than similar MHC. Third interesting thing is that virus has evolved to avoid MHC. So they can actually infect the host and kill the host. In addition, tumor can escape the immune detection by downregulating the MHC1. However, our immune system has evolved in a way that if the MHC1 downregulates, the NK cell can detect those cells and can kill. The fifth interesting thing is that MHC1 is present in neurons too. Previously, it is thought that the neurons are immune privileged site. However, the MHC1 are detected in the neuron and help for the neural development and plasticity, not just immunity. So thank you for watching. Stay tuned for the next video.